Hello, I am Mitiku and I am making this video for assignment number 3 of the HiSat tutorial. So this particular assignment is on differential expression analysis. Essentially, the assignment has two subcomponents, the differential expression analysis and data visualization, or visualizing the output from the differential analysis. So um, this is the paper or the tutorial paper on HiSat 2. So basically we are on procedure 7 here. So it's just the differential expression analysis. So for this, the first step should be loading the relevant softwares, package and libraries. And then once we load the essential softwares, the next step will be loading the phenotype data for the samples. And essentially the phenotype data contains information about our RNA-seq samples. So this file looks like yes. So it tells us about the gene ID and the sex, male versus female, and then the population from which the samples were collected, like the Nigeria and the Great Britain. Um, so once we load the phenotype data, the next step will be um, just reading uh, the the expression data, or just the transcript abundance level. So these transcript abundance levels were estimated or calculated by the string type from our assignment number 2. So we need to use the output of our assignment number 2 as an input into the assignment number 3. And then there is a step to filter or to remove the low abundance genes. So we will be keeping only those genes having a variance of greater than 1. So which means those genes having a variance less than 1 will be removed from our analysis. And the next steps will be identifying the transcripts and genes that show statistical significant difference between groups. Once those statistically significant genes and transcripts are identified, then the next step will be just to add the gene names and gene IDs to the transcript data frame. And then there's also another step to sort the results based on their p-value. Just uh, uh, putting the genes or the transcripts in ascending order based on their p-value, from that means from the smallest to the largest value. And then finally just writing the results into uh, the specific directory. And once we are done with the transcript, I mean the expression differential expression analysis, the next step will be just visualizing the output or the result. So there are a few steps to visualize the result, and the final output should look like this. So what I did is I just write the code, starting from installing the package up to the visualization. So I just check this code uh, just by using the Arrow Studio, and then it just works. But for the assignment, I will be running each of these codes from the terminal. Um, so just to say a few words about the structure of the code. So just as some introduction here about the purpose of the transcript and the date and the person who just write. And so after downloading or loading the essential package and libraries, the next step will be um, to load the phenotype data. The phenotype data is just an information regarding the RNA-seq samples. And then after that, the next step will be just reading the expression data that were calculated by the string type from basically from our assignment number 2. And there is another step for filtering or to remove the low abundance genes. As I mentioned before, we will be keeping only those genes having a variance greater than 1. After that, there are two steps. Basically, they are the same, and um, the purpose here is just to identify the transcripts or the genes that show statistically significant difference between groups. So these genes should be identified, and then after those genes and transcripts that showed statistical significance difference between the groups are identified, the next step will be just to add gene names and gene IDs to the result. Uh, and there is also another step, which is just sorting the results based on their p-value from the smallest to the largest. So the transcripts and the genes will be uh, put in ascending order based on their p-value, and finally just write the output to a specified directory. So after finishing, until this step, um, we were just doing the differential expression analysis, and then starting from this one, uh, the following few steps are um, uh, the necessary or the essential steps to visualize the dataset. So um, before I start the running ad, let me show you the yeah, let me show you the output from our previous assignment. That is the transcript abundance levels estimated by uh, the string type. Looks like this one. So we will be just using this as an input. So let me okay. Uh, I need to change the directory to this one. So first I need to call R, and then after that I need to install the essential um, package and libraries. Okay, it may take some time. Yeah, it's done. So then I need to call the bioconductor. Is bioconductor dot org slash bioclide dot r. Yeah. Okay. So after that, basically we can also call each library separately, like, but let's have them run at it, all of them at a time. Okay. So I would say no. Okay, then we need to load each library. I prefer to go one by one. then the next one will be this one. Okay, let me try just running everything at a time. It should work. Yeah, which one? This one? Yeah, library. Okay, so now I just load the necessary software in library and package. So now I'll be just loading the phenotype data. So this is just the directory where the phenotype data is saved. So I'll be just running here. Yeah, so we are safe, and now I'll be just reading the transcript abundance data from the string type. So just copy and run here. Yes, it's reading. Yeah, so the next step is just to filter um, in order to remove the low abundance genes. So as I already mentioned, so we'll be just keeping only those genes having a variance greater than 1. Okay, yeah, it just filters. And then so we just give this name just fail. We can have a different name actually, but just to indicate that this one is just the filtered one. So the next one, the next step will be just identifying the transcript that show statistically significant difference between groups. And okay, and then the same thing for the genes. Okay, and then we'll be adding the gene names and gene ID using this code to the results transcript data frame. Actually, yeah. And then these two steps, codes will sort the result based on the p-value. So the genes and transcripts will be ordered in ascending order based on their p-value. That's from smallest to largest. Yeah, it's done. And this is just the final step, just to write the gene and transcript results into the specified directory. Um, so I will check and will show you whether the results are properly written to the specified directory. And then the next step will be um, data visualization. So this step is just an optional step, but it's basically it's important because it makes the plot pretty. So just copy the code and run here. Yeah, so this is just the specified plot area. Okay, yeah, here it is. Okay, this one here. Okay, so then plot this code just used to plot the distribution of gene abundance measured as FPKM values across samples and colored by sex. We just define the color this step. So I just copy and paste here. So yeah, here it is. So this is just the result. Yeah, I think it's basically the same with the one shown in the paper. Okay, and then okay, I can minimize this one. Yes, and then the next will be just making plots of individual transcripts across samples. So I just pick sample 6. um, yeah, yes, so they are colored female by male. Yeah, nice. And finally, this one. And I don't know why I just tried this in the R, but I was not able to get the same um, plot as shown on the paper. Yeah, yeah, it's a bit different, but I couldn't find why this is just different from the one shown in the paper. Yeah, but generally the assignment looks like this one. So uh, essentially, if one just ran uh, 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 this code from R, the same result will be obtained. I just checked that before. So this is so what I have to say about the assignment number 3. And thank you so much.